Yeah.
Great. Okay, wonderful. Today, I really appreciate the opportunity to present some of the ongoing work we have ongoing at Scripps Research, and really to talk with such a really wonderful group of people organized today. So thank you to Daniel and all the organizers for this. I wanted to tell a little bit about a story about kind of what we are doing of using a known drug library collection to really answer a very fundamental question, which is, can we take drugs that have safely been moved into clinical development, either as experimental drugs or even more preferred, drugs that are approved, and take them forward into a new indication or a new disease. And certainly ability, for example, to accelerate discovery from this collection to treat, and as we're now finding out, even be able to reduce hospital stay, affect the most sickest of patients from this disease, to be able to reduce the overall burden on our healthcare system, but most paramount, being able to save lives. And so, we've been called upon over the last few years as part of a collaboration with the Bill and Melinda Gates Foundation to build a library, which we call Reframe, which I'll kind of outline in the subsequent slides. And really, at the end of the day, the goal is, can we accelerate discoveries to treat COVID-19? And as we look into the future, other potential pandemics, to be able to use this as a rapid way to move things into pandemic response. So a little bit about Caliber. So Caliber is essentially serves as a drug discovery division of Scripps Research Institute. We have the ability to do early discovery, and certainly basic science is at the core of what Scripps and many other institutions around the world do to understand disease biology. And Caliber's real goal is to be able to go from those discoveries all the way from everything from kind of engineering of proteins and antibody-based therapies, including CAR T cells, and then also what I'm going to focus on today, and what we do also a lot at Caliber, is do small molecule drug discovery, which is being able to take large libraries of compounds, really use high throughput robotic technology to quickly assay a variety of different diseases, of which COVID-19 infection is one of them that we're doing right now in the lab. And then potentially to take those molecules and go through a step which is called optimization, where certainly we'd love to find molecules that already work really well and simply move them into a clinical trial. Sometimes we're not so fortunate, and we actually have to optimize the molecules in terms of their safety, pharmacology, and optimizing their drug activity, which I think has been outlined through several of the talks this evening. And then finally, the most important part, which we've added recently, is to go into IND-enabling studies, being able to make sure these molecules are safe enough to move into clinical development, and then doing proof-of-concept trials. And so Caliber now has 5 different drugs, both small molecules, large molecules, and CAR T cells that are in clinical development. And 2 of those programs are actually what I'm going to call drug repurposing, which is being able to take drugs that are already being used for other indications and move them into a new disease. So
go ahead.
In the context of what we've done with building a drug repurposing library, this was a collection of molecules we built about in 2018. We've run many screens against this drug collection. And in fact, this library contains about over 13,000 small molecules. As you can see from the left-hand side here, many of these, a little bit more than a third of these, are actually approved drugs. And many of them are investigational new drugs, which means that they're in clinical development right now for other disease indications. And as we're now kind of learning in the world of kind of next generation sequencing and high throughput genomics, is the understanding of how drugs that may work on one or more targets could be used across multiple different disease areas and disease indications. And we're already starting to see that. I mean, there's already been a lot of reports of repurposing drugs. One of the most common ones that's been reported in the mainstream media is around the use of quinolines and anti-malarial drugs as being potentially improving in terms of COVID infection, as well as a variety of other compounds that are used as antivirals for other disease indications and being able to show activity against COVID-19. So we use high throughput robotics, as I mentioned, to do high throughput screens and find interesting hits, and then move those into either direct repurposing, which we can use the drug directly, or we can move those into an optimization to make a drug that actually is better for the new indication that we're using. The key of all of this is these molecules we're starting off with are deemed to be safe enough to move into human trials. So the amount of time it takes to optimize the molecules and move them into drug trials is much, much shorter. We can save several years in many cases, or in some cases 10 or more years, by using molecules that are already been safe to be in humans. And certainly if they're approved molecules, we would be able to directly repurpose them into a new disease indication. As I said, we built this library with the Bill and Melinda Gates Foundation, and one of the most surprising and amazing parts of this collection is, in fact, only half of these molecules are actually available commercially, meaning if you had a research lab and you wanted to screen this, you could only access about half of this collection. The other half of the collection, we actually synthesized each drug by each drug, put them together in this collection. So there's a tremendous amount of drugs, potential drugs, that are now available by people to be able to access these molecules and be able to repurpose them for the application of COVID-19. And in fact, what we've done is, before I go there, tell you two examples of where we've done this. We've started 2 clinical trials in the last year and a half as collaborations with the Gates Foundation, where we've been able to do direct repurposing. One of them is a molecule called Arnab, which is a GOLD1 complex used as a historical rheumatoid arthritis drug. We found it to be very active on tuberculosis. In fact, since we were discussing about how tuberculosis infects many, many different tissues in the body as the previous speakers, one thing that's very interesting is that Arnab and GOLD1, particular GOLD1 complexes, have activity against many other viruses and pathogens. Interestingly enough, highly active against tuberculosis, and is currently, we took drug material that was available in the US and in Europe and moved it into a TB trial in South Africa that's currently running. In addition, and probably even more useful for the concept of COVID-19, is we found a molecule that's marketed as an anti-leprosy drug called clofazimine that's marketed at no cost through public service from Novartis Pharmaceuticals. And we found that not only does it have activity against leprosy and against that bacteria that everyone knows of, but it also has activity against cryptosporidium, which is the second largest cause of childhood mortality from diarrheal infections, second only to rotavirus. And so in that case, we were able to take a pathogen that very little drug discovery had been done. We were able to go just in 9 months to go from development of a high throughput assay, which I showed here, screen the entire collection, and move it directly into a clinical trial that's now being run in Malawi for a part of the world that's endemic with childhood diarrhea caused by cryptosporidium parasites. So an antibacterial, in that case, being able to be directly repurposed into an antiparasitic. And we've been able to go, as I said, in 9 months from developing a screen, running the screen, and then being able to initiate a clinical trial. So again, the aspirational goal is to be able to do this with COVID-19. I'm very happy to say that we've actually shared this compound collection with nearly 15 groups throughout the world. One of the groups that contacted us in the early days of this infection was in the 3rd week of January in Hong Kong University, along with collaborators here in San Diego at Scripps Research and Sanford Burnham Institute. We were able to provide the reframe collection to that group. They were able to take some of the first patient isolates from patients in Wuhan, run a screen, and we actually now have primary hits, which we're actually testing right now in dose response to be able to determine which drugs may actually be potent there. Not surprisingly, in that list is many molecules that people have already described as being potential drugs for COVID-19, but there are also a tremendous amount of new chemicals and new potential mechanisms of action that could also be effective against COVID-19 infection. So we're very excited about taking those hits and moving them, as Margaret mentioned, in parallel to animal trials and animal studies, be able to move them potentially into clinical trials and to be able to benefit patients who so desperately need something for this very terrible infection. The other really wonderful part of this, as part of the Gates Foundation work, is the only thing that we ask in return. We provide the drug compounds for free. We actually make them into plates so various folks can screen it. It's one thing that's been amazing in the last 2 decades is that you don't really always need fancy robots to discover drugs. In fact, many academic labs and small labs have the tools to be able to screen 10,000 drugs. That's become very commonplace. In fact, we also have a nice example of malaria, vivax malaria infection, where we actually, instead of being able to bring the parasite to actually be screened in our labs, we actually sent a person and some equipment for 3 months to Mae Sot in Thailand, which is a high area of dormant malaria infection of hypnozoites, and actually were able to run a reframe study there in just in a short period of time using dormant parasites from patients who were affected in Thailand. The only upside for all of this is to make this annotated and open access data portal. We put all of the data that's generated from these screens. Again, we provide the compounds for free. All that we ask for people to do who are screening the collection is make the data available in real time. We have all of this information that not only combines for these 13,000 plus molecules, all the screening data we have, how does it perform against a variety of different assays? In this case, how does it compare to other viral assays like Zika and Lassa infection that we've already tested, but how does it look across a wide variety of different assays? We put all of that data along with all the drug information, meaning what companies are involved in this, what was it originally developed for, what is the potential way that the drug works, what disease indications is it used for, chemical structure, and most importantly, we put all the information of how one can access this material from folks who have already synthesized the molecule, who can provide it on scale, hopefully, and scale enough to be able to move into a clinical trial like we did with clofazimine and Olanzapine. The real aspirational goal here is that the molecules are safe and we know how to make them, and so we can quickly move them into clinical development as a direct repurposing activity. And all of this is available, as the theme was throughout the presentation today. Make all of this actionable data in real time for folks to use for this global pandemic. We've been able to use a variety of different groups to do this. I mentioned, really the goal here is we've run over 125 screens to date. As I mentioned, the stories of direct repurposing. We have many more stories like that where we can use an old drug for a new indication. And as I said, we now have several examples where we've initiated clinical trials. And again, this only happens through a global hub of global health drug discovery that's been initiated from Scripps. And then most importantly, this slide is just to show, just in the last 2.5 months since this infection has come to bear throughout the world, we've been able to establish collaborations with some of the best labs in the world, not just those at Scripps Research, but across the world who are trying to affect this infection. In fact, I mentioned University of Hong Kong earlier as one of the first groups who's actually able to run a high throughput to a medium throughput screen on reframe. And we now are engaging a variety of different groups throughout the world to really bring to bear this collection and its ability to apply to SARS-CoV-2 or COVID-19. And again, the real goal of this is really being able to help patients as quickly as possible with some of the best antiviral labs in the world and drug development groups. Thank you very much.
Awesome. Thank you, Arnab. So one question is, we all hope that you and others will find drugs quickly that work, but then there's the question of resistance. We know from the HIV world and others often takes sometimes a cocktail. What's your thought on the need to combine and how resistance might emerge?
Yeah, no, so that's a great question, and similar to what Margaret, I agree with her sentiments on this as well. One thing we have already started doing is to start running combination drug trials. So where one could actually take a drug that has shown some promise as a single agent, for example, remdesivir or chloroquine or T705, I can name a few that folks have already described, and also include some of the work that people have done in, for example, in silico modeling, where we think certain mechanisms of action should be not only efficacious against the virus, but potentially also be able to be used to be combined so we can limit resistance of the virus, which will most certainly happen over time, and that's an inevitability. So the hope is also that the collection is small enough where one could actually do not just rational combinations, but more importantly do unrational drug combination trials where we could take a drug and put on top of it all these other molecules from the collection and see what may potentially synergize, or mechanisms, for example, directly affecting the virus, for example, like remdesivir does and other polymerase inhibitors, combine it, for example, with a host factor that may have an effect on just general viral replication in host cells, and be able to combine those so that one could actually be able to come up with a combination which could potentially also limit the ability for resistance to emerge for this infection.
Got it. And last question, given you're at Scripps, a major academic institution, your postdocs and grad students aren't in the lab right now. What are the implications for research in general when you necessarily can't be there
Yeah, no, that's a great question. So, at the end, I think, given that we're in San Diego and in California right now with the shutdown that's here, we are actually just exclusively working on things related to COVID-19. So I'm here at my house. Many of the people involved are remote, and really we have two biosafety level 3 labs that are open where folks are going in. So science has slowed down. The overall effects of this virus is not only the medical impact and the hospital impact and the impact on society and economies, it also has an impact on the ability to progress science all throughout our work. And so our ability to really be able to tackle this problem as a global health community is critical for us to be able to bring up all of the advances that have gone on even outside of this particular infection.
Well, fantastic. On behalf of all of us looking for new cures, thanks for all your work and collaborations. It was good to see some of your work on 60 Minutes last night as well.
Right.
Any takeaways you would just give for folks in the academic world working on similar problems that you've learned in your work that could translate to others?
Yeah, I mean, my sense is that, really this drug collection and really for us, it's kind of funny for me to say as a chemist who, makes a living on optimizing drugs to get to drug candidates, this drug collection and the ability to share this without any strings. The intellectual property is owned by the people who are running the screens. The only thing we ask is that the data be made publicly available. In that sort of an environment, I think it's critical to get as many different labs around the world to test these molecules and bring their data together and see what we actually have as actionable items. I think that's critical. And in fact, right now, in the world that we live in, democratizing drug screening and democratizing biology and being able to not have a biologist have to know a chemist who makes molecules for them of value for an actual screen or to potentially use as a treatment. A physician or a biologist doesn't need to know a chemist to do this. And providing this collection from them is critical for us to be able to cut out a lot of this process that slows down the development of drugs and be able to more directly be able to get them to patients.
Great. Well, Godspeed on your work, and thanks again.
Thank you.
All
Thanks.
right.